Important questions, that you might well ask. First, but one, why does reverse transcriptase use AZT even though it has the wrong 3' end? You know, that is, didn't I just talk about how enzymes are super specific and they bind their substrates just so? AZT has the wrong shape. It doesn't look like T. It's got that big N3 sticking out instead of an OH. Well, it turns out that reverse transcriptase is not very picky about its substrates. That is, RTase is not selective about the 3' ends of its substrates. That is, it's not bright enough to tell the difference. So it can't tell AZT from normal T. Number two, what about the human DNA polymerase? In the say it's in the patient, right? Because we have DNA polymerase. Why don't we use AZ? And the answer is our DNA pol is more picky, but it sometimes uses AZT by accident. And that leads to some side effects of AZT treatment. So it turns out that human DNA polymerase, for the most part, says, oh, wait, that 3' end of that molecule just doesn't look right. I'm not going to use that. Something's wrong here. I'm not going to use that substrate. Every once in a while, it goofs up and does use it. And then you get these chain terminations that messes up DNA replication. So in certain cells, their DNA isn't replicated properly. And that leads to some symptoms which are similar to that of chemotherapy, which is that rapidly dividing cells get messed up. Like sometimes you get sort of hair loss and nausea and a variety of other nasty side effects, although they're usually relatively minor. The last thing you might ask is, what about mutant? Go away. Right. Oops, it looks like I'm running low on batteries. Yikes. It's going to excuse me for some technical difficulties for 1 second. I don't know why Windows is causing me this trouble. Now we're back in business. What about mutant HIV? Sorry. And that is, so if the mutation makes reverse transcriptase more picky, so it doesn't use AZT, that means this strain is going to be resistant to AZT. So one of the constant problems, because HIV in any patient is a constantly moving target, it's always evolving in response to whatever your body tries to do it. It also can evolve in response to AZT. So that if you've got a population of HIV in your body, you take AZT, most of them are going to get killed off. However, any one of them that has a mutant reverse transcriptase that's mutated in just the right way so it doesn't use AZT anymore, so maybe it's got some side chain that pokes in there and prevents AZT from coming in, who knows what, then AZT is not going to poison that virus, and that virus is going to be able to replicate. And so you're going to take AZT, and it will kill 99.9% of the virus in your body, but that 0.01 or 0.001% that still survives, well, they're going to come right back up. And so one drug at a time, it turns out, is a losing battle against AIDS, because you become resistant to drug one, and then you take another drug, and then it becomes resistant to drug 2, et cetera, because this has such a high mutation rate. And so, again, this was the first line of treatment. The second role was to treat with what they called a cocktail of many nucleoside analogs. And it's hard to be resistant to all at once. So what they now treat with is a cocktail of several, 2 or 3 or 4 nucleoside analogs, and there's essentially no chance that a mutant HIV is going to be able to be simultaneously resistant to all of them. So you knock the HIV down big time, and then your immune system can wipe out what's left. But the worst possible treatment would be to treat with drug 1 until it stops working, and then treat with drug 2, because now you're going to have resistance to drug 1 and drug 2, and then drug 3. If you take the drug serially, it's a disaster, because you end up with a strain at the end that's resistant to all 4 drugs. Whereas if you hit with all 4 at once, the chance of something being resistant to all 4 at once is almost 0, so you wipe them out. Questions about that thing before we talk about one other kind? Yeah. Exactly. So what he was asking is, so nucleoside analogs are going to bind to reverse transcriptase and the DNA polymerase, but much less well to DNA polymerase. So all of these things will make you somewhat sick, but they make the virus sicker, if you like. That's right. Yeah. So has that resulted in a functional cure? There's one more kind of drug I'm going to talk about. My understanding is at this point it is quite difficult to say that a person is ever cured of AIDS because there's those latent viruses, the dormant ones, right? You can knock the virus down so low that it essentially hasn't come back. I don't know if they've done studies about taking people off antiretrovirals, because who's going to want to volunteer for that? But it looks like you can come close to a cure when you use a rather extensive cocktail of things. Let me talk about one last kind of anti-HIV drug, HIV protease. This processes HIV's proteins into active forms. I'm sorry, it's supposed to be B. I don't know what I'm doing with my letters here. Maybe you should just pay no attention to the man behind the curtain. Theoretically, AZT was supposed to be A, maybe. Yes, reverse transcriptase was the target A. So this is technically B. I think I'm on target. So the last step in HIV is sort of ripening, right? The virus is formed. The proteins have to be processed. Remember, they made as one monster protein and chopped up into little bits. And so that HIV protease, the enzyme that does that chopping up, is unique to HIV, and so therefore is a totally excellent target for anti-HIV drugs. So you use what are called protease inhibitors. They don't block all proteases. You need those for digestion, just HIV protease. And these are drugs designed based on the structure of HIV protease. Right? And they bind to the active site and prevent enzyme from working. So these are a lot like the amylase inhibitors you guys saw in lab, that they bind to the active site of the enzyme and they block it. And so the active site would like to do its job and it can't because the inhibitor is bound there. So that's where the starch blockers, I don't know, in some lab sections the starch blockers worked, in some lab sections they didn't. That's probably not surprising. The protease inhibitors are much more effective, and they're have been tested by the FDA, and they inhibit the HIV protease. And so if you can inhibit that enzyme, then the virus doesn't mature. Therefore, virus doesn't mature into active form. Therefore, it can't infect cells. All right? So that's another really promising target. And the interesting thing about this is there's two different kinds of inhibition. When we talk about inhibiting reverse transcriptase, like up above with AZT, we're actually not preventing the enzyme from doing its job. We're letting it do its job. It's just kind of planting a bomb in the DNA's chain. Right? That is, it does its job. It does its job just fine. It puts a nucleotide, and then you can't continue. So it's like sticking some gum in the works. But the enzyme still works fine. HIV protease inhibitors actually prevent the protease from doing its job at all. They bind to its active site, block it up, and the enzyme can't do its job. And as a result, the proteins aren't formed properly, and so the virus can't infect cells. Yeah? So one suggestion is, couldn't you just destroy someone's immune system and basically starve the HIV, right? And then you try to put it back later. Yes, presumably, well, there's two things. As long as you're sure that HIV only infects immune system cells, and I believe there are some other ones it infects. And the other thing is it would be enormously risky, because basically you'd be giving yourself sort of full-blown AIDS, hoping to hang on and not get sick in the meantime before you put your immune system back. I think so. There are theoretical reasons for not suggesting that, although it's not a bad idea, and practical reasons as well. But given it's the target, you might imagine something like that working. Other questions? Yeah. Yep. Did I? No, I'm sorry, isn't. I didn't mean it that way. Let me put it this way. That's this line up here. No, if the mutation makes reverse transcriptase more picky, so it doesn't use AZT, then the virus is resistant to AZT. Yeah. So yeah, you're right. That would. I'm a little addled today. I don't really know why, but I'm not that addled. What I meant is the virus is resistant to AZT. Because you're right, that would be nonsense. I'm glad I didn't write something that crazy. All right. Let me just say one before I completely lose my marbles here. If you take A plus B plus your immune system, reduces HIV levels significantly. And the question, which is still unanswered, is can you eliminate HIV completely? The answer is probably, but it's not really clear. Actually, I should say it probably this way. It's probably not. But the jury's still out. There are some specific cases, like there's strong evidence for like healthcare providers who get like a needle stick, like they're working on a patient who's HIV infected with a needle, so there's HIV on the needle, and they get stuck with a needle. They take an extremely heavy dose of a protease inhibitor, as well as nucleoside analogs, heavy course of that sort of thing for a short term, basically knock this thing down before the infection can get going at all. And it seems like people who treat that can take a short course of those drugs and are then remain HIV-free for the rest of their lives. So early on, it seems like you can clear it. If you're fully infected, it is not clear that you can ever make it go away. My understanding is that currently you take these drugs for the rest of your life, that you can't ever say that it's totally gone away because of those dormant ones. Questions about that? Yeah. Do you say sick for the rest of your life? The answer is no. You are, because you can knock the HIV down so low that your immune system has a normal function. So you have the side effects of the medication, so you have the side effects of the medication, but that's all. Right.